it's Sarah, and today we're working on macromolecules, which are very large molecules, and so I wanted to record a video so that you had that in addition to your text. There's a macromolecules practice worksheet that you should be working on while we go over this to make sure that you can recognize the pictures of the different kinds of molecules. But in general, macro means big, just like micro means small. So macromolecule is a really big molecule. There are four major kinds of macromolecules inside of you, and most of these come from your food. And then you use those molecules from your food and rearrange them through different kinds of chemical reactions to make the molecules that are parts of your cells inside your body. So the four big kinds, we got carbohydrates. Probably know what carbohydrates are. Things like sugars and grains have a lot of carbohydrates in them. Lipids include fats as well as some other things. They tend to be hydrophobic. Oil doesn't mix with water very well. You've got proteins. Protein also comes from your food. And most of what's in your body besides water is proteins, lots of different kinds. And then we have nucleic acids, and there are two of those. They're DNA and RNA. So the first thing we got to talk about is that most of these macromolecules are made from small subunits called monomers. Mono means one. And you put those monomers together to make polymers. So in this picture, we're just using a circle to show a single monomer. So we got one circle, and if we put them together to make many, that's a polymer. So poly means many. All of your four kinds except for the lipids are made of subunits called monomers that we stick together to make polymers. So the way that we do that is through a reaction called dehydration synthesis. And if you get dehydrated, it means you don't have enough water. You've lost water. So what happens is these monomers and polymers are strung together, and at the end of them they will usually have these groups that have an H or an OH. And you should know by now that water is H2O, and so if you take the OH and the H and you stick them together, that will make H2O and make water. So what happens is if we want to stick this short polymer together with this unlinked monomer, we put the H from one of them together with the OH from the other one. We make a water molecule. The water molecule goes away. And now, right here, where the H and OH were, we have a bond between those two monomers. So we have just built this thing bigger. We started out with 3 monomers together and 1 monomer by itself. We added that one on, and we have a longer polymer. This happens anytime you're trying to build a new carbohydrate or a new piece of DNA or RNA in your cell. Anytime you're trying to build a new polymer in your cell, like if your cell has to grow or make a new cell because you are growing or repairing a wound or something like that, then you're constantly having to do this dehydration synthesis reaction. So remember, dehydration, we're taking away water. Synthesis means making something bigger, so we're putting things together. The opposite kind of reaction is called hydrolysis. And hydro often means water. Like if you think of hydropower, people use water behind dams and going through turbines to do power. Lyse means break. So here we're going to add a water molecule and break a bond. So it's exactly the opposite of dehydration synthesis. We take a water molecule and we stick an H from that water molecule on one side, and we stick the other H and the O on the other side. And that breaks this bond that we originally had in our polymer. So we're basically breaking a monomer off of it. This happens anytime you digest your food, because your food molecules are too big to get inside of your body cells by themselves. So you have to break them up into little pieces and then bring the little pieces inside of your cells, where they can be used for energy or to build new macromolecules. So now we got to go through the different kinds of macromolecules. So first we have carbohydrates. So you've probably heard of starch. It's in things like potatoes and bananas. It's a big carbohydrate. Sugars as well. The monomers of carbohydrates are specifically called monosaccharide. So saccharide means sugar. So monosaccharide is one sugar. And if you have a whole bunch of them together, that's called a polysaccharide. So if we're going to put a bunch of monomers together, monosaccharides, to make a polysaccharide, we're going to have to use a dehydration synthesis reaction. So the way to recognize a monosaccharide, you're going to have to be able to recognize pictures of them on a test. You won't have to draw them, but you do need to recognize them. They will usually look like hexagons or pentagons. So hexagons have 6 sides, pentagons have 5 sides. So they're usually going to look like either a hexagon or a pentagon. And they're going to have multiples of CH2O. So remember, CH2O, if there's no number after the letter, that means there's just one of them. So we could say C1H2O1. So this means this thing has 1 carbon, 2 hydrogens, and 1 oxygen. And if we're going to multiply that by something, let's say we multiply it by 5, then 1 times 5 we get C5, 2 times 5 that would mean H10, and 1 times 5 we get O5. So this would be a multiple of CH2O, so that would be a monosaccharide. The most common monosaccharide multiplies this by 6, and that's called glucose. It's C6H12O6. When people talk about your blood sugar, it's glucose is the main one that's in there. The other 2 monosaccharides that you get from a lot of your food are fructose and galactose. And they come from different kinds of food. People usually call fructose fruit sugar. They have the same formula as glucose, C6H12O6, but they look different in shape. So like glucose is a hexagon and fructose is a pentagon, but they still both have 6 C's, 12 H's, and 6 O's. So when 2 things have the same atoms but different structures, we call them isomers. So glucose and fructose are isomers of each other. The main purpose of monosaccharides is to break them down for quick energy. It's the easiest thing for your body to break down when you need energy, which is why it's good to eat something with carbohydrates right before you do a really intense workout. So here is a picture of a few ways to draw glucose. Usually in your body, it's in this third form. It looks like a ring. So we said that they usually look like a pentagon or a hexagon. And it shows you there, and you could count the numbers of CH's and O's. And remember, glucose should have 6 C's, 12 H's, and 6 O's. So we could count 1, 2, 3, 4, 5, 6 C's. For H's, we got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and where am I missing? Oh, this one, 12. So we got 12 H's, and if you count, you'll get 6 O's as well. So if you want to put 2 monosaccharides together, remember that you do a dehydration synthesis reaction, which involves taking away some H's and O's and making them into water, and then you're going to put 2 things together. So the prefix for 2 is di. So we call it a disaccharide if we have 2 monosaccharides put together. So depending on which ones you put together, you'll get a different disaccharide. So if you stick glucose and glucose together, you can see that there's still an extra little O in between them right there that we didn't use to make the water. So that's still going to be there when we stick the 2 together. So now we have maltose is made from 2 glucoses. You don't have to memorize which disaccharide is which, but know that if there's 2 monosaccharides connected, that it's a disaccharide. Here we got another one. Glucose and fructose put together makes sucrose. This is the sugar that you buy at the store. So if you buy a bag of sugar, it's going to be sucrose. But you're going to need to know, because books and stuff will start doing this, they'll start being lazy with drawing molecules, and you'll start seeing something that looks like a zigzaggy line like this thing at the bottom. Whenever you see something like that where it doesn't label something, if there's an edge or a corner, there's really a carbon there. So we can assume that all these things are carbons. And if you remember from our previous section, carbon has atomic number of 6, which means it has 2 electrons in the first shell and 4 in the second shell, and it needs 4 more to get full. Since it needs 4 more to get full, it will make 4 bonds. Right now, you can see that this carbon right here only has 2 bonds. It's bound to the carbon on the left and the one on the right. So it needs some more bonds. If they didn't draw all the bonds in for your carbon, you can assume that it is actually attached to some extra hydrogens in order to make 4 bonds. So since this carbon right here has 2 bonds already, it needs 2 more to hydrogen. And since the one on the very end only has 1 bond to something else already, it needs 3 more bonds. So we'll add 3 hydrogens on there. This is just a warning because you're going to see pictures like this. This is what it means. Edges or corners are carbons. If the carbon's not attached to 4 things, there's really some hydrogens in there attached to it. So here is a simplified drawing of glucose that only shows some of the things. So if you're trying to count the numbers of carbons and hydrogens and oxygens, look for any corner or edge that doesn't have a label, and that's going to be a carbon. So we can see right here, this corner is going to be a carbon, this corner is a carbon, this one's a carbon, this one's a carbon, and this one's a carbon. Now we needed 6 carbons, so we got 1, 2, 3, 4, 5, and the 6th one is actually shown, and it's up here. I think it actually has all the hydrogens and oxygens on this particular picture, but if it didn't, you know, if your carbon didn't have 4 bonds, then there's some extra hydrogens attached to it. So if you put more than 2 monosaccharides together, then we call it a polysaccharide, because poly meant many. The polysaccharides are used to store energy, and then if you want to actually use that energy, then you're going to have to break them up into monosaccharides. And they're also used for structural support, so they can be pretty strong. And there are a bunch of different polysaccharides, and most of the common ones have only glucose, but they arrange the glucose in different ways. So here's a couple examples. So you can see starch on the left and glycogen on the right. Both of these are used for storing extra energy. So the starch is used to store extra energy in plants, and the glycogen is used to store extra energy in animals, including humans. So you can see all of those little tiny hexagons in there. Those are your glucoses. So both of them are just glucose, only glucose. But the thing that makes them different is that the starch has glucose mostly going in lines with only a few branches off of the main line, whereas glycogen has a lot more branches to it coming off of the main string of glucoses. But for instance, since humans are animals, if your blood sugar gets too high, then you release the hormone insulin, and insulin tells your cells to bring the glucose inside the cell and put them all together to make glycogen and store it for later. And then if you're exercising a lot and using up all of your sugars for energy and you need higher blood sugar, it's too low, then those cells that store the glycogen will break it up with hydrolysis reactions and then put those individual glucoses back in the blood so that your muscles can grab them and use them for energy. So then our third and last major polysaccharide you need to know is cellulose. Cellulose is for structural support. So it's really supportive and strong, and it makes up the wall of plant cells. They have cell walls. when you get stringy celery stuff stuck in your teeth, that's cellulose. And wood is also made mostly of cellulose. It's also glucose only, just like starch and glycogen were, but the bonds are different from each other. And you might or might not know that humans can't digest cellulose. It's what we call fiber. And when you eat it, it goes all the way through your digestive system and comes out in your feces. So it's not something that you can absorb for energy. But it's kind of weird because it's made out of glucose, and we know that we absorb glucose for energy. So the reason we can't absorb it, as you can see right here, in starch, the glucoses in starch have this OH group at the bottom of every single starch, or every single glucose in the starch. And in cellulose, the OH group is actually in a slightly different placement. And so you can see that in some of them it's down at the bottom and some of them it's up. It alternates. And it turns out that that creates a difference in the bonds. You can see the oxygen in this first bond. The bonds going to it are going up, and in this one they're going down. We have enzymes in our bodies, in your digestive system, that can break the bonds between the starch glucoses, but they can't break up the cellulose glucoses, which means that those glucoses stay attached and they're way too big to get into your cells, which means they stay in your digestive system all the way till the end and come out in the feces. This is your dietary fiber. Which means, since it's not getting absorbed by your cells, that it's not providing calories or extra energy for you. Here's a picture of cellulose, and you can see that it makes these long, stringy chains that make the plant cell walls. So, major functions of your carbohydrates, we already said that monosaccharides like glucose are used to get fast energy. You can break them down, and breaking down those bonds releases energy that we use to make ATP, which is the name of our main energy molecule in the body. So easiest thing to break down for energy is monosaccharides like glucose. Another thing that we use them for is for identifying your cells. So we have these things called glycoproteins on the outside of our cells, and they're a protein attached to a chain of glucoses usually. So glycose is the old name for glucose, and it's attached to a protein. And you can see right here that this one looks like a chain of glucoses in a Y shape coming off the outside of that cell, but people have different shapes of glucose chains coming off of their cells, different shapes of glycoproteins. And your immune system recognizes the specific glycoproteins that you are supposed to have, so that if some other living thing that's not part of you gets inside your body, it's not going to have the same glycoproteins as you, and your immune system is going to know that since it doesn't have the right ones, it's an invader. Now this ends up being super important for things like blood types. That's the reason why you can't give some blood types to other blood types, is because they'll have a different glycoprotein and your immune system will see it as an invader. It's also important for organ rejection. So when you're trying to find a match for an organ transplant, they need to have similar glycoproteins to you. And also for fighting pathogens or germs. Basically, your immune system needs to be able to recognize that something doesn't belong in this body. It doesn't have the right ID tags or glycoproteins. So that's the end of carbohydrates. Let's talk about lipids now. Lipids are the ones that don't form polymers. They mostly have the elements carbon and hydrogen, so we call those together hydrocarbons. And if you remember from our last section, O and CH, the C and H are neighbors, and neighbors make nonpolar bonds. They're going to share their electrons equally, which means they're not going to have any partial charges, so they're going to be nonpolar and hydrophobic, so they don't dissolve in water very well. Oil and water don't mix. So there are three major important types of lipids in the body. They are fats, phospholipids, and steroids. So we're going to go through those three. So starting with fats. Fats have two parts. They've got a glycerol and they've got fatty acids. And this picture right here shows you what a fatty acid looks like. Now, you may have noticed in the periodic table there is no element called R. R just means there could be any other thing attached to that. It means that part varies between the different fatty acids. It's usually some long chain of carbons and hydrogens, but they could be different lengths. So, like, if you remember our shortcut way of drawing things, we can just do a long chain like this. It looks like a zigzag, and at every corner, in reality, there's a carbon. And if this carbon doesn't have enough bonds, it should have some extra hydrogens until it has 4 bonds. So a fatty acid could look like this, or you could have one that's longer or shorter, or one that's got some double bonds in it. But that R just means there could be different things there. So we said a fat has two parts. It's got the glycerol and the fatty acid. Fat's also called a triglyceride. So the glycerol part of it looks like this. It's got some carbons, bonds all the way around them, and on one side the bonds are all to OH, and all the rest of the ones are just to H. So this is the glycerol. And we already said that a fatty acid looks like it's got this C with a double bond to an O and an OH, and then it's got something hanging off the end that's just a bunch of carbons and hydrogens. So a triglyceride has a glycerol plus 3 fatty acids. So we're going to have 3 of these fatty acids. We'll make one with a double bond. And we'll make another one. So these right here are supposed to be 2 lines. I know my drawing's terrible. That's supposed to be a double bond. So it's a carbon double bonded to another carbon there, which means that this carbon right here has 2 bonds to another carbon and 1 bond to this one down here, which means it only has room for 1 hydrogen. So we got a triglyceride, which is a glycerol plus 3 fatty acids. So FAs are fatty acids. And they're going to be connected through dehydration reactions, which remember means we got to find 2 H's and an O, connect them together to make water, and the water goes away, and let's erase. So we got rid of the 2 H's and an O, and now there's an O left in the middle there. That connects. So dehydration, we take away water. Synthesis, we connect them. So let's do that for all 3. Take away 2 H's and an O to make water, and whatever's left will get connected. So now we have a triglyceride that is glycerol connected to 3 fatty acids. Kind of ugly fatty acids, but you see what I mean. Here's a prettier picture of it. So you can see the glycerol is on the left side in gray, and the 3 fatty acids are on the right side in yellow. Now, I mentioned that they could have double bonds between 2 carbons. So if I was going to create a double bond between these carbons, like right here, right now this carbon now has 5 bonds. It's got 2 to that carbon, and then 3, 4, 5. Which means one of the hydrogens has to go away. And same with this one, it's now got 5 bonds, so one of the hydrogens has to go away. So if you have a double bond between 2 of the carbons, then that fatty acid cannot hold as many hydrogens. So we say that is unsaturated. It's not completely saturated with hydrogens. It doesn't have the most possible hydrogens for that numbers of carbons. But if there are no double bonds between carbons, they're all single bonds, then it's saturated. So the top fatty acid here is unsaturated now that I drew this double bond, and the second one and the third one are both saturated. If there's even one unsaturated fatty acid in the whole entire triglyceride, then the whole thing is called an unsaturated fat. So this one's an unsaturated fat now that I made that double bond. Sometimes people say it's monounsaturated if there's only one double bond, or polyunsaturated if there's more, and so you might have noticed that on food labels, but I won't test you on mono or polyunsaturated, just whether it's saturated or not. So here's a description. So the saturated have no double bonds between carbons, unsaturated have at least one double bond between carbons. So here's a picture of a saturated fat. You can see all three of them have no double bonds between the two carbons. Saturated fats tend to come from animals most of the time. It's like butter has some, it comes from milk, which comes from cows, so that's got some saturated fats. And they tend to be solid at room temperature, things like butter or meat. Here is an unsaturated fat, so you can see that there's a double bond between two of the carbons. And that double bond makes the whole thing unsaturated, and these tend to be liquid at room temperature, and usually they come from plants or fish. So things like olive oil is unsaturated fats mainly. There are two kinds of unsaturated fats, cis and trans. And you can see right here that they both have a double bond between the carbons. But in the trans, the double bond does not cause the zigzag to change its shape, the general shape it's making. Whereas in the cis, the double bond makes it bend. These cis double bonds are found in most natural products that have unsaturated fats, whereas the trans ones are usually created through a manufacturing process called partial hydrogenation, where they're trying to make a fat more solid, and so they'll take an unsaturated fat and break some of the double bonds and make it more like a saturated fat, so it gets it more solid. And these trans ones tend to be very bad for you. It's really hard for your body to break them down, so they stick with you for a long time. So as far as fats and how good they are for you, it's generally best to get most of your fat from cis unsaturated fat. And some saturated fat is okay, but too much of it can lead to heart disease. And the trans fats, people say you should try to avoid these as much as possible. You don't need any trans fats in your diet. So you might think, okay, why the heck would you even try to make a trans fat, hydrogenate fats in the first place? Well, it's because it makes it more solid, which is useful for things like margarine or shortening, and it also makes them last longer. So a lot of like fryer oils at fast food restaurants have trans fats in them. So now that we've talked about what fats look like and the difference between saturated, unsaturated, and cis and trans fats, we need to talk about what they're good for. So if you remember, polysaccharides, one of their functions was for energy storage, but they tend to be for sort of a medium-term energy storage. It takes usually about 20 minutes to use up all of your glycogen as stored energy if you're exercising. And so fats are then your long-term energy storage. So if you used up all your energy from glycogen, then you'll break down fats to release extra energy to make more ATP. We store our fats in cells called adipose cells, or sometimes people just call them fat cells, and it's mostly used for energy storage, but also to some extent for insulation and cushioning. So next we got to talk about a second kind of lipid called a phospholipid. So remember, there were three important kinds of lipids. We had fats, triglycerides, we had phospholipids, and we had steroids. And the fat or triglyceride was made of three fatty acids connected to glycerol. So the phospholipid is kind of similar, except for instead of three fatty acids connected to glycerol, we only have two of them. And in the place where the third fatty acid would normally be, instead we have something called a phosphate group. So you can see on here, it's a little bit of a simplified picture, but we've got our two fatty acids right here. These are the two fatty acids. In this particular one of them is saturated and has no double bonds between carbons, and the other one is unsaturated and has that double bond that makes it bend. But there are phospholipids that have two of them that are saturated or two that are unsaturated. There's a bunch of different kinds of phospholipids. Then up at the top here, connected to the third carbon in the glycerol, we have our phosphate group. And a phosphate group looks like a P, which is a phosphorus, surrounded by some oxygens. And you'll see that there's some charges on this. There's a negative charge on that oxygen. There's a positive charge on this thing up here. So the phosphate part of this is charged, which means it will dissolve in water, which is polar and also charged. Charged things dissolve in other charged things. So this part is hydrophilic, and people will often call that the head. So in this really simplistic drawing down here, that's the part with the phosphate, that's the head, and it's hydrophilic. And then these tails down here, all those corners that aren't labeled are carbons, and they're connected to four things, so they need some invisible hydrogens. So these tails are hydrocarbons. They're made of hydrogen and carbon, and if you remember from our O and CH again, carbon and hydrogen are neighbors, so they're nonpolar. They don't have any charges. And so that makes them hydrophobic. If they have no charge, they're not going to dissolve in water, which does have a charge. So those parts, we call them the hydrophobic tails. Those parts will not dissolve in water. So phospholipid has got the hydrophilic head with the phosphate part, and the hydrophobic tails, which are like the fatty acid part. These are super important in biology because they make up cell membranes and membranes around organelles. So this is a picture of a cell membrane, and you can see that there are two layers of phospholipids. So if we were to outline a single phospholipid, I'm really bad with drawing with the mouse, but there's one phospholipid, and it's got the head and it's got the two tails. And they arrange themselves so that the head is next to something that contains water. And on the second layer, you can see the head here is also next to the watery liquid inside of the cell. And the tails hide out in the middle layer where there is no water, because they're hydrophobic, water-fearing. So cell membranes made of two layers of phospholipids. Sometimes we call it a phospholipid bilayer, because bi means two, just like di also means two. So our third and last kind of lipid we're going to talk about is a steroid. And you've probably heard of anabolic steroids that people sometimes take illegally for sports. So a steroid is a type of lipid. There are a number of different kinds of steroids, but they all have a common structure. They all have 4 fused carbon rings, and I'll show you in a second what they look like. But a few examples: cholesterol is a steroid. It helps stabilize the cell membranes. It's in with the phospholipids in many cases. A lot of hormones are steroids, especially the sex hormones like testosterone and estrogen and those. And when people take steroids for sports, for muscle growth, it's often a synthetic version of testosterone. Vitamin D is a steroid too. It's actually really similar chemically to cholesterol. So here's a picture of a steroid. This particular one is cholesterol, and they always have 4 fused carbon rings. So you can see the yellow rings over here, and the reason we call them carbon rings is because see all those unlabeled corners? Those are carbons. So there will be a carbon at each one of those edges there. And if it doesn't have 4 things connected to it, you can draw in hydrogens for the rest. So this hydrocarbon right here is connected to this carbon, which is 1 bond, 2 bonds, 3 bonds, and 4 bonds. So all steroids are going to have those 4 rings like that. The thing coming off the edge could be a little bit different, or some of them might have more double bonds in their rings than others, but they're all going to have the 4 fused carbon rings. You should be able to see one of these on your macromolecule practice worksheet. So that's our 3 kinds of lipids. Lipids are the ones that don't have monomers or polymers. We just have 3 important kinds, which are the fats, triglycerides, we have the phospholipids, and we got the steroids. So remember there were 4 kinds of macromolecules. We had carbohydrates, lipids, proteins, and nucleic acids. So we've done carbs and lipids, now we're on to proteins. Proteins are the most diverse of the 4 types. There are tons and tons of different kinds of proteins. And they are the major thing that makes you different from other people. You've probably heard people say that that's DNA, but it turns out DNA is it's the instructions for how to make your proteins. So your DNA is different from other people's DNA, but it's different, and what that means is that it causes you to make different proteins than they have. So the monomers of proteins are called amino acids, and there are 20 different kinds of amino acids. Some of them are essential, which means you've got to get them from your food, and other ones you can make in your own body. The 20 different amino acids have a part called an R group, and the R group is different for each of the 20. So this is what an amino acid looks like in general. All 20 amino acids have an N connected to some H, which we call the amino group. All 20 amino acids have this carboxyl group, which looks like the thing that's on the end of a fatty acid. Fatty acids also have a carboxyl group. And all of them have a carbon in the middle with an H on the bottom. This part up here, the R group, remember R is not an element. It means there could be multiple different kinds of things up there. So here is a list of all 20 amino acids, and you can see that the part on the bottom, this part, is the same for all 20 of them, and the part that's in the white box, that's the R group, which is different for each of the 20 amino acids. So this one up here, glycine, has a very small R group, it's only a hydrogen, whereas something like tryptophan up here, which you might have heard of because it's in Turkey, has a really large, bulky R group. So the difference in R groups is what makes those amino acids different from each other. I'm not going to make you memorize which one's phenylalanine and which one's tryptophan or any of that. You just have to be able to tell when you look at a picture, is this an amino acid or not? And to tell whether it's an amino acid, you need to know what that bottom part looks like. It's got an N with some H's and a C with some O's. So when you put amino acids together using dehydration synthesis, you're going to get a polypeptide. So polypeptide is the polymer made from a bunch of amino acids. Sometimes a protein has just one polypeptide, and sometimes a protein has more than one polypeptide. So in this picture down here, these are meant to represent a string of amino acids, which is a polypeptide. So this one right here on the left has one polypeptide, and this one on the right, you can see there are 4 polypeptides together, and that makes one protein here. So this protein has 4 polypeptides. This protein on the left only has one polypeptide. Different proteins have different numbers of polypeptides. This is showing how you make the bond to form the polypeptides, and it's just dehydration synthesis like before. So Each of these things in the box is an amino acid, and the R group is what's coming out at the top. So here we've got two different amino acids connected together, and we're adding a third one. And to do that, we take the OH and the H, make them into water, which goes away, and now we have a bond between our amino acids. So there are lots of ways to draw proteins. We've got one on the left here, but ultimately they twist and turn and fold up based on like positive and negative charges in the R groups sticking together in different ways. Here's our space-filling model over on the right that shows you the large protein that ultimately forms. The shape of the protein determines its function. So here's an example right here. We've got on the left an antibody protein made by your immune system, and this helps to attach to pathogens, disease organisms. And then on the right we've got a protein from a flu virus, and we can see that the shape of the antibody matches up pretty well to let it attach to the flu virus protein. So the shape of the protein is determined by which amino acids are there in which order, and your DNA is what determines that. So DNA tells you which amino acids to stick on to make the protein, and the protein shape determines its function. So even a really minor change in one amino acid of a big long protein can really mess it up sometimes and cause diseases. So if you have a mistake in just a single letter of your DNA, it could cause you to put the wrong amino acid on your protein, which could give you a disease, for example, sickle cell anemia. So sickle cell anemia is based on a mutation in a gene in your DNA, and that gene tells you how to make the hemoglobin protein. Hemoglobin is the main protein that carries oxygen in your blood. So if you have normal hemoglobin with the right amino acids in it, the hemoglobin protein looks pretty round and fills up your blood cell and makes it nice and round so it can fit through blood vessels easily. But if you have this DNA mutation that causes your hemoglobin protein to have one wrong amino acid, then the hemoglobin proteins get long and thin and clumped together and make your blood cells look like a sickle or half moon kind of shape. And these tend to get stuck in blood vessels and cause clots and pain, and people with sickle cell anemia often have to go to the hospital pretty frequently for complications. So it turns out that even if your protein has all the right amino acids, you can sometimes mess up its shape due to something like changing the pH, changing the salt or ion concentrations, changing the temperature. All of those can potentially cause your protein shape to unravel, which we call denaturation. So a denatured protein has unraveled and no longer has the correct shape or the correct function. So here's a picture. You can see the normal protein is supposed to fold up into this shape on the left, and if you were to heat it up too much or put it in the wrong pH, it could get denatured or unraveled, and now it no longer has the shape that it needs to carry out its function. Sometimes proteins can be renatured if you put them back into the normal conditions, but not always. So if you think about like cooking an egg, you're not going to be able to get that cooked egg back into its raw form. So its proteins have been irreversibly denatured in that case. Though I think read about somebody did an experiment recently and they figured out what conditions were necessary where they could actually get a cooked egg to be like it was raw again. But it wasn't something that you could do regularly in your house. So since there are 20 different amino acids, 20 monomers, that means there are tons of combinations that you could make to make a functional protein. And so there are lots and lots of different shapes and lots of different functions for proteins. If you dry out your cell, most of what's left is going to be proteins. So some of the different things proteins can do. A lot of them are in your cell membrane. So some of them are attached to glucose chains to make like those little glycoproteins for recognizing which cell is foreign versus part of your own body. Some proteins your DNA wraps around them and folds up and tightens before your cell divides and make more cells. Some proteins can turn genes on and off. So here we can see part of a gene, well, multiple genes up here, and this is a repressor protein, which is like a switch that turns these genes off so that they will not be used to make other proteins. There are other proteins that can be activator proteins that activate certain genes to then make the protein that they code for. This is why, even though all the cells in your body have the exact same DNA instructions, you can have some cells that do different things. Like a blood cell looks a lot different from a neuron in your brain, which looks a lot different from a liver cell. It's because different genes in their DNA are activated by different proteins. Antibodies are a type of immune protein that attaches onto pathogens that invade your body, and those antibody proteins make it much easier for you to fight and kill those pathogens. There are proteins in your cell membrane that help things to move in and out. So this moving black thing right here is a protein that is acting a little bit like Pac-Man to let these molecules in and out of this cell. We've got proteins that transport oxygen, like hemoglobin. We've got proteins that act as hormones. So for instance, insulin is a hormone and it's also a protein. And when you eat a lot, normally you would release insulin inside of your body from your pancreas, and that insulin is going to travel through the blood and give a message to your liver cells and your muscle cells to bring glucose inside the liver and muscle cells so that it's no longer high in the blood. People with type 1 diabetes can't make enough insulin and their pancreas isn't working correctly, and so they can get insulin shots to replace that insulin, and then the shot is the message to the cells saying, take in the blood sugar. Proteins can act as enzymes. An enzyme is a protein that speeds up chemical reactions. A lot of the chemical reactions in your body, including things like dehydration and hydrolysis, would take thousands of years to work without the protein. So imagine if you eat a meal and it's going to take 1,000 years to digest your food, to get it into your cells for energy. Obviously, you're going to be long dead before your cells get the energy from that food. And so the enzymes are going to help break up that food through hydrolysis much faster so that your cells can get the food inside for energy and survive. So here's an example of an enzyme. The purple thing is an enzyme. Enzymes almost always have the letters ASE at the end of their name. So this one is sucrase. Usually the name of the enzyme relates to what it does, so this enzyme helps us to break up or hydrolyze the sugar sucrose. So enzymes always have a specific shape in them called the active site, and the sucrose in this case is going to fit into the enzyme at that active site, and the enzyme can squish it in certain ways to help it do the reaction faster and break up that sucrose into two monosaccharides, glucose and fructose. So there are other enzymes that do different reactions. They don't all break up sucrose. Some of them will break up proteins, some of them will build proteins. All the reactions in your body pretty much are going to be catalyzed by enzymes. So our last of our 4 major types of molecules is the nucleic acids, and there are 2 kinds, DNA and RNA. The main purpose of DNA is to give you instructions for how to make proteins. So it's like a coded instruction for how to make proteins. RNA is going to be involved in the actual process of making the proteins. So the DNA is like the main instructions, and the RNA helps to actually use the DNA instructions to make the proteins. So here's kind of how it happens. In the nucleus of a cell, which is the major large organelle, that's where the DNA is stored. And then a piece of the DNA is going to get used or copied onto a piece of RNA. and then that RNA is going to come out of the nucleus into the rest of the cell, which we call the cytoplasm, and then that RNA is going to get used to make the protein. So here you can see that the RNA is getting read by this ribosome organelle, which we'll talk more about later. And as it gets read, we're connecting these amino acids in the correct order based on those instructions to make our protein. So this whole process, don't worry too much about it. We're going to go into detail about it in a future chapter, but that'll be a long time from now. But the main thing to know here is DNA is the instructions for how to make the proteins, and RNA is involved in the process of actually making the proteins. So nucleotides are polymers. The monomers are called nucleotides, and the polymers are called polynucleotides. The monomer, the nucleotide, has three parts. The three parts are a nitrogenous base, a pentose sugar, and a phosphate group. So we've seen what a phosphate group looks like already. It was a P with some O's around it. So you know what that looks like. Pentose sounds like pentagon, which has 5 sides. So it's going to be a sugar that's got 5 sides, and we know that sugars usually have 5 or 6 sides. The nitrogenous base, you can guess that it's going to have some nitrogen in it, some N, and we'll see what it looks like. So here are some nucleotides. It's got the 3 parts. So here's the sugar, and it's a pentose sugar, it's got 5 sides. Here is the phosphate group, and here is the nitrogenous base. And this is a really simplified picture, so you can't see it, but there's really some nitrogen around the edge of the nitrogenous base. And we connect these nucleotides together to make a polynucleotide. So over on the left, we've got a polynucleotide made of 4 nucleotides. So here is one of the nucleotides, and you can see that that nucleotide unit is repeated 4 times to make this polynucleotide. So there are a bunch of different nitrogenous bases that could be in your nucleic acid. So your nitrogenous bases, we often label them by their first letter, and DNA has 4 different kinds. It's got the C, the T, the A, and the G. And RNA also has 4 different kinds. It's got the C, the U, the A, and the G. So the T is only in DNA, and the U is only in RNA. But these are our nitrogenous bases. So if you're looking for parts to put together to make a nucleotide on your practice macromolecule worksheet, you might want to take a look at these, because one of those parts to put together is going to be a nitrogenous base. You can see they've all got N in them. So every single one of these has some nitrogen in it. These are our pentose sugars. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. So you can see in here that the DNA has the deoxyribose sugar and the RNA has ribose sugar. And you might notice that the only difference between them is right here. The ribose sugar has an extra oxygen, and the deoxyribose, there's no extra oxygen there. So they call this deoxyribose because deoxy sounds like less oxygen, so it's got less oxygen than ribose. That's where it got the name. So these nucleotides in the DNA are going to coil up and make a spiral form called a double helix. So there are actually 2 polynucleotides that connect together, and then they twist to make this double helix. And the sugars and the phosphates are on the outside of the double helix, and the nitrogenous bases pair up in the middle. The A is going to pair with the T, and the G is going to pair with the C. So here's a picture. So you can see this is very simplified, but the sugars and the phosphates are going to be this part at the edge, like the banister on a spiral staircase. And the nitrogenous bases are the part in the middle. So we can see right here we got an A with a T and a C with a G. So, and you might notice also they are connected by hydrogen bonds. Hydrogen bonds always have that sort of dotted line to show that they're weaker than some of the other types of bonds. And this allows us to actually split open the two polynucleotides of DNA when we are copying it. So this picture is actually showing the DNA being copied, where the strands at the top are being split open and we're making new strands to copy them down at the bottom. So the order of the nitrogenous bases, the A's and T's and C's and G's, is the DNA code that tells you how to make the protein. So for instance, if your DNA said AATGCTG, that would make a different order of amino acids, different protein than TATTGGA. So that order is what differs between people and between different species, and ultimately determines what different kinds of proteins you're going to make and what kinds of looks and personality traits and all that kinds of things that you have based on your genetics. So that's the end of macromolecules. Hopefully that was somewhat interesting. This will definitely build up. We'll return to lots of different things that involve these kinds of macromolecules in our next chapter. Feel free to send me questions if you get lost on anything, and don't forget to do your practice worksheet to make sure you understand this stuff.